Welcome children. In today's video number 3, cell cycle and cell division, I am going to deal with the cell division mitosis. We know where mitosis occurs. It occurs in the somatic cells or body cells, and the different phases of mitosis are prophase, metaphase, anaphase and telophase. But to get started, we have to start with the initial phase, that is interphase, and we have to focus only at certain organelles, namely the nuclear membrane, which is represented by a red color circle, nucleolus represented by a blue dot inside the nucleus. You can place it anywhere inside the nucleus. Chromosomes and the centrosomes. You all are well aware of the structure of the centrosomes. It consists of two cylindrical structures called centrioles, which are placed at right angles to each other. Okay, so and we have to just ignore the other organelles. Only focus on these four structures, namely the nuclear membrane, which is represented by a red circle, and I will continue with these colors till the end. Nucleolus, which is represented by a dot anywhere inside the nucleus. Chromosomes being the first stage, it is represented like thread-like structures, and centrosomes comprising the centrioles, which are at right angles to each other. Now here the orientation, only take care of the orientation of the centrioles or the centrosomes. That decides how you have to move about, how you have to proceed with the different stages. Now I would like to draw your attention to this. In different texts it is shown in different ways. If you refer different biology texts, you can see cell cycle, cell division and the orientation of the cell. So now itself I am going to explain the orientation I have taken, and you can understand the orientation with respect to the centrosomes. I place the centrosomes on towards the right hand side. Okay, so that indicates my orientation. In some texts you can see this, I am going to orient it like this, so you can see the centrosome at the base, in some towards the left hand side and in some towards the upper portion. I am taking orientation towards the right hand side. So we will proceed like this way. Halfway we cannot change the orientation. So only take care of the location of the centrosome. Place it to the right hand side in the middle at the equatorial plane. Okay, that should be the orientation. The others and all, it's being a square. Okay, all squares I like, or all rectangles I like. Circle also. Nucleolus, place it anywhere inside the nucleoplasm, and the chromosomes, as I explained, you will understand. Okay, so that's how we are going to proceed. So you need not be bothered of endoplasmic reticulum, Golgi apparatus, plastids, vacuoles, nothing. Only focus on these organelles. So let's get started. The first phase is the interphase. To start off with mitosis or meiosis, we have to start with the initial resting stage called the interphase. Though it's called resting stage, it's the stage where the cell is getting prepared for cell division. The first point you have to emphasize is the nuclear membrane. Look at the nuclear membrane. It is intact. The nuclear membrane is intact. The second point, look at the nucleolus. That is also intact and prominent. Third, centrioles, which is outside the nuclear membrane. That is also intact. And fourth, look at the fate of the chromosomes. How are the chromosomes? The chromosomes here are described as elongated threads. When they are elongated, thread-like structures, and when they have to be accommodated in a small spherical structure, definitely it gets entangled. Can you identify the number of chromosomes in this interphase stage? Can you easily identify? No, we cannot identify. But actually there are 4 chromosomes. I am going to indicate chromosomes with yellow color. So there are 4 long threads, 1, 2, 3, 4. To keep these threads, fine threads in a small circle, it gets entangled. So in this context, it is referred to as, in the interphase stage, chromosomes which are like thread-like entangled mass on network, it is called as chromatin threads. It is referred not to chromosomes, it is referred to as chromatin threads or chromatin. So, this is reworded. Chromosomes in the interphase stage is called chromatin. So, recap of interphase. The nuclear membrane is intact. So, you are drawing a circle which I have indicated with red. The nucleolus is intact. I have given it with a blue dot inside the nucleus anywhere. Centrioles are intact. Here I place it on the right hand side. So, my orientation is always placed at the right hand side, outside, just outside the nuclear membrane. And the chromosomes, how are they? They appear as fine threads, and these fine threads are called chromatin. Okay, and these fine threads, because they have to be accommodated, they become like an entangled mass or network. Clear? Now we will move on to the interphase. As we are moving from one phase to the other phase, what are the changes occurring? Mainly the changes are occurring in the shape of the chromosomes. The chromosomes, as we move from one phase to the other phase, undergoes a process called condensation. What is condensation? It becomes shorter, 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 and along with that becomes thicker and thicker, and accordingly the shape changes. Okay, that is very progressive. And also we are going to see changes occurring in the nucleolus, nuclear membrane and the centrosomes. So, as we move from phase to phase, you can see what are happening to these four structures. Prophase. So, as I am dealing with each phase, I am drawing. The drawings are not ready-made. So, we know this is a cell, so I drew the cell boundary, not relevant here. Next, the nuclear membrane. Now, look how I am drawing the nuclear membrane. The nuclear membrane are drawn like a dotted circle. What does this indicate? The nuclear membrane is getting disintegrated. Then the nucleolus, which I took blue color, okay, there I drew very prominent spot to show nucleolus is intact, but here I am going to give a just light dot to show that even the nucleolus is also disintegrating. I hope you can see this. Okay, very lightly you have to show a spot anywhere inside the nucleus. So, it is indicating the nucleolus is disintegrating. Next, look at the centrioles. I have represented the centrioles here, as two cylinders. Now, what happens is they split and move to the opposite poles. The correct word, they replicate and move to the opposite poles. So, in your diagram you must show with arrow we are on their way to the opposite poles. Clear? So, about the centrioles replicate and move to the opposite poles. Then about the chromosomes. As I told you, the process of chromosome is undergoing condensation. Now, here the chromosomes condense from thread like structure, condense and become rod like structure and slightly thicker. So, I am making the nib slightly thicker. See, we can see now there are 4 threads. Now, the 4 threads became 4 rods. So, by the process of condensation. So, what happens to the chromosome? The chromosomes condenses. When it condenses, what it becomes short and thick. And here it is visible like rods, rod-like structures. Clear? And you see that you scatter it. Don't align it in this specific orientation as I did. It's at your discretion. You put 4 rods at any points or any location within the nuclear membrane. Clear? So, you don't look into the orientation and just copy it down into your notebook. You can place 4 rod like structures anywhere in the nuclear membrane, provided it does not lie on a straight line. So, here and there, okay, with different orientation. So, once again a recap of what happens in prophase. 1. The nuclear membrane and nucleolus disintegrates. So, how is that indicated in the diagram? As dotted. The nuclear membrane as dots and the nucleolus as a light patch. Second, the centrioles replicate and migrate to the opposite poles. See how I migrated? I send those to the opposite poles with arrows indicating the poles. And lastly, the chromatins, it was thread-like, becomes rod-like by condensation. Clear? Now you can count the number of chromosomes. We had 4 chromosomes. Moving on to the next phase, that is metaphase. Metaphase, again as usual, I am going to draw the boundary which represents the plasma membrane. Now, very important, what is the first event? The nuclear membrane and nucleolus has already disappeared. Now, the disintegrated nuclear membrane and nucleolus has completely disappeared. So, you need not draw nuclear membrane or nucleolus. Second point is the chromosomes. I told you chromosomes, the ongoing process condensation, it further condenses and becomes visible as X-shaped structures, X-shaped structures. Okay. So, when you talk about X-shaped structure, what comes to your mind? The metacentric chromosome. It need not be metacentric, it can be submetacentric, acrocentric, telocentric. I am going ahead with my diagrams being the metacentric chromosomes. Okay. So, it further condenses. When it condenses, what happens? The chromosomes become thicker and now it becomes X-shaped structures. X. And here you can see that constriction, primary constriction, what we have learned, that demarcates the chromosomes into the different types. What is the primary constriction called? Centromere. And a chromosome has 2 members. The 2 members are the chromatids. So, this is one chromatid, this is the second chromatid. So, you need not call one chromatid, second chromatid, these are called sister chromatids. So, the chromosome will have 2 sister chromatids attached at a point called the centromere. Clear? Now, here also the orientation can change. My diagram I have given this orientation. Clear? Anyway, it looks, it's X, and not only this, very importantly, these chromosomes align at the equatorial plate. Okay, it not only becomes shortened and X-shaped structures referring to the metacentric chromosome, they align at the equatorial plate. And lastly, what happens is the spindle fibers. Third point, spindle fibers arise from the centrioles at the 2 poles. We saw the centrioles by now has reached the 2 poles, up and down, and they give rise to the spindle fibers. So, with these descriptions, I am going to draw. Nuclear membrane, nucleolus, I need not draw because they have disappeared. Now, I am going to draw the 4 chromosomes, and definitely they have to be aligned at the equatorial plate. So, 4 X's at the center, at the equatorial plate. And very importantly, demarcate the centromere. Clear? So, the chromosomes condenses further to form X-shaped structures. X-shaped in bracket you have to definitely indicate metacentric chromosomes, because it need not be metacentric, can be telocentric or submetacentric. I am going with metacentric chromosomes. So, the chromosomes condenses to form the X-shaped chromosomes and they align at the equatorial plate. So, that one structure or one event. And next event is we have the centrioles at the poles. I made very prominent. At the poles there are the centrioles. They give out what? Spindle fibers, fine spindle fibers. And these spindle fibers that arise from the centrioles grow and attach each chromosomes where? At the centromere. So, once again the centrioles which are at the poles, they send out spindle fibers, and these spindle fibers grow and attach the centromere of each chromosome. Okay. So, this is the structure of metaphase. Clear? Only 2 events, and so only 2 structures you have to indicate in your diagram. Need not draw nuclear membrane or nucleolus because they have totally disappeared. Next, what very important event, the chromosomes further condenses and become X-shaped structures like the metacentric chromosomes and align at the equatorial plate. Meanwhile, what happens? The centrioles at the poles, at the two opposite poles, give out spindle fibers, and these spindle fibers attaches with the chromosomes. All the spindle fibers attaches with the chromosomes. Where? Not at the arms, at the centromere. Clear? Next we will move to the anaphase stage. Now, as usual, I am drawing the boundary which represents the cell wall or the plasma membrane. Now here what happens? A very, very important event occurs. I told you the centrioles at the poles had given out spindle fibers. Now these spindle fibers contract. See, these spindle fibers contract, and when they contract, they pull or they split the centromere of each chromosome. Thereby the chromosome gets separated into sister chromatids. The sister chromatids get separated away and move to the opposite poles. Once again, here the important event is by the centrioles. The centrioles have given out the spindle fibers. These spindle fibers contract. So where is the pressure applied? It is applied on the centromere. So when it is applied on the centromere, what happens? The centromere connects the two sister chromatids. What happened to the two sister chromatids? They separate from each other and move to the opposite poles. See? Look at this. Here pressure is applied. This is representing the centromere, which is attaching the two sister chromatids. Upper one sister chromatids, lower this one sister chromatid. Pressure is applied here and here. So what happens to the sister chromatid? They separate from one another. I will show you this with the help of a video clipping. Clear? Just focus at the chromosomes aligned at the equatorial plane and look at the centrioles. Now look here, the orientation different. So it started with the centriole at the top. That's why now the centriole at the right hand side. Okay. Now I am going to on the clipping. See, they held at the centromere and moved to the opposite poles. So they went to the opposite poles. I hope you have understood anaphase with this video. So this is the diagram that depicts anaphase, where the chromatids separate from one another because of the splitting of the centromere and they move to the opposite poles. Clear? So that visual video should be effective here. How this happens? Okay, pressure is built up on the centromere splits. When the centromere splits, only the chromatids, both sisters chromatids separate from one another. Next we have the telophase. In telophase we have to indicate both early and late telophase. Okay, so the state of the cell in early telophase. Okay, this is representing early telophase. Okay, now what happens here is we saw the sister chromatids went to the opposite poles. Okay, this is the state we saw as a result of anaphase. Now here you have to remember the spindle fibers have disappeared. The spindle fibers that were produced by the centrioles disappear totally. Okay, and now is the reappearance of the nuclear membrane. Nuclear membrane reappears around the sister chromatids at the two poles. Now these two sister chromatids are no more referred to as sister chromatids, they are called daughter chromosomes. So nuclear membrane reappears, even the nucleolus also reappears. You can put the spot anywhere inside, such that you can see two nuclei in one cell. This is depicting only early telophase. Now I am going to make the late telophase, a replica of this diagram. So the late telophase, the difference between early and late is with respect to the chromosomes. the chromosomes do not further condense. Now it becomes opposite, it elongates and become fine threads. Where it's so much it gets elongated and becomes fine threads. 1, 2, 3 and 4 chromosomes here, 1, 2, 3 and 4. So, this is the situation of the late telophase. So, the difference between early and late is with respect to the chromosomes. In early telophase, the chromosomes as seen as such split chromatids moving to the opposite poles, and look at the orientation, these are inverted V and these are V shaped because the pressure built on the centromeres, in that direction they are pulled. Okay. So, they look like this. Okay. And here the very important event, major event is nuclear membrane reappears around the daughter chromosomes. Again further we are not calling sister chromatids, we are calling it daughter chromosomes. So, nuclear membrane reappears, nucleolus also reappears. Now, in late telophase what happens? These chromosomes not condense, they elongate and they form an entangled mass called chromatin threads, going back to the past. Now, what happens? Next, if I proceed here, what happens? It is now karyokinesis over. Okay, we got 2 nuclei. Now, it is followed by cytokinesis. Now, cytokinesis in animal cell and plant cell, it is different. In animal cell, what happens is a constriction. If I am showing with another kind, a constriction is formed in the plasma membrane. See, constriction is formed in the plasma membrane between, and this constriction is between the 2 daughter nuclei. This constriction deepens and deepens. See, I am making it deep, deepens and deepens, and finally divides the cell into 2. Whereas, so this is the case in animal cell. Whereas in plant cell, it is different. It is not by constriction. A cell plate is formed. See, one cell plate is formed between the 2 nuclei within the cytoplasm, and this cell plate grows outwards. Okay, it is going to grow outwards, growing outwards and outwards, and it cuts the cytoplasm into 2. So, finally, we get 2 daughter cells. With the nuclear membrane intact, nucleolus intact, centrosome intact, and the chromosomes in the form of fine threads called chromatin. So, the correct description of the interphase. So, once again I am going to reiterate the cytokinesis occurring in animal cell and plant cell. In plant cell, what happens? A cell plate is formed in the cytoplasm between the 2 nuclei, and it grows outwards and divides the cell into 2. Whereas in animal cells, the reverse, from outside. A constriction is formed. A constriction is formed in the cytoplasm. See, constriction is formed which deepens and deepens and deepens and divides the cell into 2. Okay. And we have finally 2 daughter cells with a description of the interphase. The nuclear membrane is intact, the nucleolus is intact, the centrosome is intact, and the chromosomes are in the form of elongated threads. I hope you have understood up till cytokinesis. Now, so once again, in plant cell, a cell plate is formed in between the 2 nuclei which grows outwards, dividing the cytoplasm into 2 cells. Whereas in animal cell, a constriction is formed between the 2 nuclei outside in the plasma membrane which deepens, dividing the cytoplasm into 2 cells. So, we got the result of mitosis, 2 daughter cells. So, a recap of all the events here. Ready? We started with interphase, and how we describe the interphase. I told you to concentrate only at 4 organelles, namely the nuclear membrane, nucleolus, chromosomes and centrosomes. So, in the interphase stage, what is the state of all these 4 organelles? The centrosome is intact outside the nuclear membrane, the nucleolus is intact inside the nucleus, the nuclear membrane is also intact, and the chromosomes, what is the state of the chromosome? They appear as fine threads, and these fine threads are called chromatin threads. That is interphase. Now, prophase. The nuclear membrane disintegrates. See, the nuclear membrane is disintegrating, the nucleolus is disintegrating, the centrioles, daughter centrioles are on its way to the poles, see, moving to the opposite poles, and the chromosome condenses to form rod-like structures, rod-like. So, I am referring to these chromosomes to rod-like chromosomes. And remember, these rod-like chromosomes are scattered in the within the nucleus. Next, metaphase. What is happening? The nuclear membrane has totally disappeared, the nucleolus has totally disappeared, and the state of the chromosome, it has further condensed and become egg-shaped, referring to metacentric chromosome, and the align at the equatorial plate. So, that is the description of the chromosome. And second point, the centrioles by now has reached the poles and they give out spindle fibers. These spindle fibers attaches each chromosome where at the centromeres. Next, anaphase. In anaphase, it is an action taking place. The spindle fibers from the opposite poles contract. When they contract, what happens? The centromeres split. When the centromeres split, the chromatids, sister chromatids separate from one another and go to the opposite poles. Okay, this is the state of anaphase. Okay, moving to the opposite poles, the sister chromatids. Henceforth, they are not going to be referred as sister chromatids, they are going to be called daughter chromosomes. Next, we have the telophase. As I told you, telophase, we have the early and the late telophase. The early telophase is the chromosomes are just like the anaphase chromosomes. Surrounding the chromosomes, what reappears? The nuclear membrane reappears, the nucleolus reappears, we have the centrioles already at the poles, and the state of the early telophase chromosomes, it is still condensed. Late telophase, what happens? They elongate to form chromatid threads. So, I reiterate, the difference between early and late is with respect to the chromosome. The other characters are the same. So, once again, what are the events occurring in telophase? Reappearance of nuclear membrane, reappearance of nucleolus, and what else? The chromosomes. Initially, the chromosomes are condensed like anaphase. Later, when switching over to the late telophase, they become elongated. When they become elongated, they become the chromatin threads. Then finally, we have two nuclei in one cell. Okay, now it's time for cytokinesis. So, PMAT, that is prophase, metaphase, anaphase and telophase, indicates karyokinesis. Now it's time for cytokinesis. Cytokinesis is the splitting of the cytoplasm, dividing the cell into two. Now the way it is split is different in animal cell and plant cell. In animal cell, by constriction made in the plasma membrane between the two nuclei, whereas in plant cell is by a cell plate which is at the center. It grows outwards and cut the cytoplasm into two, or divide the cytoplasm into two. And finally, we have two daughter cells as a result of mitosis. And if you analyze the chromosome number of these daughter cells, it is 4 in number, 4 chromosomes. When you can go to the previous stage, say anaphase, you can count the chromosome 4. Because it's entangled, we can't find 4. So, there are 4. So, as said, as the difference in mitosis, how many daughter cells are formed? 2 daughter cells. And what's the chromosome number? 4 as the initial parents. We started with 4 chromosomes. Finally, the daughter cells are also having 4 chromosomes. I hope you have understood the different phases of mitosis. Okay. So, mitosis also should initiate with interphase. Though the phases are prophase, metaphase, anaphase and telophase, you have to start it with interphase. Thank you.